Hello, my name is Carrie Smoke, and today I want to talk about transmutation and the evidence for it, and whether or not transmutation of species can really occur in nature. I am an apologetics lecturer and I have a master's science degree in public health. For more information about myself and my background, please see my video called My Story. This is the fifth lesson in this series about let the evidence speak. And again, today's talk is on transmutation of species. We'll talk a little bit about DNA and then get into the idea of transmutation. Neo-Darwinists say that random mutations can explain transmutation of species. Darwin did not know about genetics and DNA. Neo-Darwinism is a revision of Darwin's ideas to include genetics and DNA. So let's look at the evidence. First, let's kind of review about DNA. This is a simple diagram showing transcription. Down here at the bottom, we have a chain that's called messenger RNA, and the triplets form an amino acid, the kind of purplish ball on top of the green transfer RNA. And this is how messenger RNA makes a protein, a very simple diagram of the process. So transfer RNA decodes messenger RNA in making a protein. Mutations can occur in the DNA letters that is represented by C, G, A, and T. This can potentially change an amino acid which make proteins. For example, ACC codes for threonine, but AAC codes for asparagine. So there can be a substitution or a mutation that code for a different protein. But instructions are needed to make proteins. The first proteins could not have originated by random mutation and natural selection without instructions. This is similar to building a house. Blueprints are needed to know how to build a house. If you look at the schematic up here, you can see a type of blueprint for a house. You can see a garage and so on, master bedroom and so on. So we have here a diagram, and this diagram tells a contractor how to build a house. Instructions are needed to specify the house that's going to be built. Let's look at it in a chemical sense at some work by Douglas Axe, a PhD in biochemistry. Proteins are initially long chains of amino acids when they are first made in a cell. Proteins must fold into a specific shape to be functional. In other words, the long chain is not functional. By folding it, you can make it functional. And you see a diagram here of a long chain on the left, and yet when it's folded, it now becomes a functional protein. The unfolded has no function at all. So let's continue looking at the work of Douglas Axe. He took a 150 amino acid sequence protein, which is a relatively modest sized protein, and made random mutations to the protein. What he found was that only 1 in 10 to the 74th power amino acid sequence for a 150 amino acid protein will fold into a stable protein. In other words, all the combinations that you could have of 150 amino acids, only 1 in 10 to the 74th will be able to fold into a stable protein. Furthermore, not all folded proteins are functional. For functional proteins, only 1 in 10 to the 77th amino acid sequences for a 150 amino acid protein will fold and be functional. Thus, the chances of a 150 amino acid protein of being functional by random mutation is exceedingly small. Instructions are necessary. So let's look at the idea of irreducible complexity, which was made popular by Michael Behe, PhD in biochemistry, in the early 1990s. What irreducible complexity is: the minimum number of parts needed for a functional system. Let's look at a mousetrap to get started with an easy example. A mousetrap consists of 5 interacting pieces: the base, the catch, the spring, the hammer, and the hold-down bar. All of these must be in place for the mousetrap to work, as the removal of any one piece destroys the function of the mousetrap. So here's a simple diagram of a mousetrap. And it's this idea here of irreducible complexity. If you take away one part, and the 5 parts are the platform or the base, the spring, the hammer, the catch, the holding bar. If you take any one of those 5 pieces away, you no longer have a functional mousetrap. So let's take it a little bit higher now to a biological example, the flagellar motor example. The flagella of certain bacteria constitute a molecular motor requiring the interaction of about 40 different protein parts. Removal of any one of these parts either renders the flagellar motor inoperative or diminished in function. Thus, according to Michael Behe, the chances of a flagellar motor occurring by random mutation is exceedingly small. Why? Because you have 40 different parts, and removal of one part makes it not functional. In fact, the flagellar motor could not have been built by random mutation in natural selection. No combination of the parts less than 40 would produce anything useful. And this is exactly the opposite of what Darwin predicted, that things should be happening by slow, gradual change by random mutation and natural selection. But we see here an example that contradicts this idea of natural selection and random mutations producing something new. Why? Because you have to have all 40 parts. Anything less than 40 doesn't make anything useful. Therefore, according to Darwinian selection, anything less than functional should be deselected, not selected for advancement. So let's look a little bit more at this. This is the flagellar motor. It's inside. So you can see the long tail, the green tail, that's outside of the organism. And then you have inside the organism this motor. It looks like an outboard motor on a boat. And that's its function, is to rotate and turn that tail very rapidly so the organism can move. Scientists have tried to refute Michael Behe's flagellar motor example and show how the flagellar motor could have evolved over time. So let's look at how they have tried to explain this. They claim that research on biochemical complexities showed that they can arise naturally and spontaneously as the result of self-organizing chemical processes. But let's think about that for a minute. The self-organizing means there has to be design and purpose. Something has to be happening to make it into something that's useful. And again, that's contrary to the idea of evolution being random and not purposeful. So self-organization is really opposite of what evolution argues. However, let's go back to the work by Douglas Axe to realize that the flagellar motor is composed of 40 proteins, and each protein of various lengths of amino acid sequence has an exceedingly small chance of arising due to random mutations. Humans have approximately 2 million proteins in their body. The longest protein in the human body is composed of almost 27,000 amino acids. So when you think about this, each of the 40 proteins that makes the flagellar motor are each irreducibly complex, according to the work of Douglas Axe. The odds of them self-organizing and forming 40 different proteins and then self-assembling into a flagellar motor is really the opposite of what evolution is about. Let's take a break and just think about this a minute and summarize so far of what we looked at. First of all, proteins are complex and they need instructions, and in our human body, the instructions come from DNA. Then just to review, Darwin says random mutations and natural selection explain the origin and diversity of life. So let's go a little bit deeper. Now we've had this talk here so far about DNA, and the evidence that we are going to look at in the next section is, can random mutations explain transmutation of species? We'll look at genetic factors inside the cell. And then, can something other than random mutations explain transmutation of species? We'll look at this. It's called epigenetics outside the cell, and we'll go into more detail to what these mean, genetics and epigenetics, in the next few slides. So let's talk about transmutations. Inside the cell, let's talk about random mutations. To make major changes in a body plan, in other words, the transmutation of species, the mutations would have to occur early in the developmental process, the embryo stage. Changes at a later stage would not have any effect on the shape of the organism, the body plan. In other words, changing one form into another form. Here's the fruit fly cycle. So you can see on the left the fruit fly and the sperm and the egg and the fertilized egg, and it starts to form the embryo, and then the embryo goes into a pupa and again produces a fly. Very briefly summarizing here. So inside the cell of random mutations, let's look at what could happen with fruit flies. Scientific experiments with fruit flies as embryos on the developmental gene regulatory network called the DGRN showed that mutant fruit flies produced deformed fruit flies that died before they could reproduce. In other words, the mutations were fatal. Thus, mutations in the early development cannot produce changes in body plans. So we can look here at the result of mutant fruit flies. On the left, you see a normal fruit fly, and in the middle, you see a curling wing. Oh, excuse me, on the right is the normal, in the middle is the curling wing, and then on the left is the short wing. So look at that there. What happens when you interrupt in the embryo stage, what's called this DGRN? It results in death or mutations that are deformities. Furthermore, mutations which are not fatal occur late in the development process are not capable of changing the body plan of a life form. In other words, after the embryo stage, any attempt to change the developmental process does not produce a change in life form. That's why scientists have focused on the early stage of an embryo. Let's continue and explain a little bit about this DGRN, again, which stands for the Developmental Gene Regulatory Network. What the DGRN does, it regulates the early cell cycle progression, sex determination, zygotic gene transcription, segmentation, and neurogenesis, among other processes, and they are composed of networks of genes and gene products that control the timing and expression of genetic information during the development of an animal, especially during the embryo period. They ensure that the developing organism produces the right proteins at the right time to service the right type of cells during the embryological development. In other words, it says at the right time, produce a heart, produce a lung, and it does it in a proper sequence so things are built together in the right order at the right time. So let's look here at the complexity of DGRNs. They look like an electrical schematic diagram as shown here. Again, you can't see all the detail, but this is like a complex system, a network of genes that are regulating the process of an embryo. So DGRNs, we have a person here, Eric Davidson in 2011, who said, there is always an observable consequence of a DGRN subcircuit when it is interrupted. Since these consequences are always catastrophically bad, flexibility is minimal, and since the subcircuits are all interconnected, the whole network partakes of the quality that there is only one way for things to work, and indeed, the embryo of each species develops in only one way. And again, I want to emphasize here that embryos only develop in one way. The idea that random mutation can change the body plan so that they morph into another organism does not work because of the DGRN. Furthermore, random mutations have no effect on the DGRNs. There is no evidence that DGRNs can be rewired. A couple of lessons ago, we talked about the Cambrian explosion, and in that talk, Professor Marshall of Berkeley proposed rewiring of DGRNs as a way to explain the Cambrian explosion when complex life forms suddenly appeared 543 million years ago. So let's look outside the cell at body plans and epigenetic information that controls what life forms look out. Epigenetics simply means outside the DNA, and it's information that is outside of DNA that controls what a life form will look like. So let's look at similar DNA in echinoderms. Here we see in the top figure the body form of 5 types of echinoderms. For instance, at the top there's a sea star, there's a sea lily, and there's a sea urchin, and so on, sea cucumber. And you can see why they're called echinoderms, derm meaning skin, and echino mean like prickly. So you can see that all of them are prickly on the outside. So they're all similar and their DNA is similar, and yet their body forms look vastly different. A sea star looks very different than a sea cucumber, yet their DNA is very, very similar. In fact, 97% similarity of DNA in sea stars, also known as starfish, sea cucumbers, and sea urchins. They have similar features but look very different. Why is this? But why is this? Outside the cell, body plans are controlled by epigenetic information. And again, epigenetic information means that something is above or beyond the genome, the DNA. So epigenetic information is information that is beyond the genes. Genes are not the whole story in how organisms develop. And this is an important point. Yes, the instructions are in the genes in DNA, but there are other factors outside of DNA, and we'll explain this in just a minute. Experiments on newt salamanders embryos as far back as 1924 have shown that organisms can develop up to a point without a nucleus where DNA is stored. That's what we mean by epigenetics. Something that is outside of DNA can influence an organism to develop. So why is epigenetic information so important? Because that's where the whole theory is going to unravel, according to Jonathan Wells in 1993. Neo-Darwinists have assumed that genes possess all of the information necessary to specify the form of an animal. They have also assumed that mutations in genes will suffice to generate the new information necessary to build a new life form of animal life. Stephen Myers, 2013. In other words, scientists have assumed that everything is controlled by DNA, but in actuality, there are factors outside of DNA that control the idea of organism and what they will look like. This is where transmutation of species falls apart. Information is necessary to build the lower level electrical components, but the same information does not determine the placement of the components on a motherboard. Additional information is necessary. So let's look at a motherboard. Here's a diagram of motherboard, and again, we're not interested in all the details, but the idea here is that to build the lower level electrical components, that does not determine the placement of the components on the motherboard. In other words, the parts do not determine where they go. Yet building materials do not determine the floor plan of the house or the arrangement of houses in a neighborhood. Stephen Meyer, again in 2013. In other words, when you're building a house, the materials do not determine where they go. There's a blueprint diagram that determines where parts go. In a similar way, DNA does not by itself direct how individual proteins are assembled in these larger systems or structures. Cell types, tissues, organs, and body plans during development. Jonathan Wells, 2001. What are examples of epigenetic information? skeletal structure, cell membranes, electromagnetic fields. These are the way to look at them. In this diagram here on the left, you see genetic information. You see that down below the word genetic information, that's is static information. It's DNA and proteins, whereas epigenetic is dynamic. For instance, electromagnetic fields. In other words, it can change. And so you have two things working to ensure that a body becomes what it's intended to be. You have genetic information with static and epigenetic information, which is dynamic. So if DNA isn't wholly responsible for the way an embryo develops for body plan morphogenesis, the idea of a body plan, then DNA sequences can mutate indefinitely and still not produce a new body plan. Regardless of the amount of time and the number of mutation trials available to the evolutionary process, genetic mutations are simply the wrong tool for the job at hand. Next quote, it follows that the mechanism of natural selection acting on random mutations in the DNA cannot by itself generate novel body plans such as those that first arose in the Cambrian period. And these are by Stephen Meyer in 2013. So let's summarize this. Making proteins requires instructions, and the instructions come from DNA. Random mutations cannot cause transmutation of species. Mutations early in embryogenesis are usually fatal. The developmental gene regulatory network cannot be rewired. Body plans and epigenetic information that is beyond the DNA control what life forms look like. Genetic mutations cannot alter what a life form looks like. Does the evidence support the idea that random mutations can cause transmutation of species? Using inference to the best explanation, what would you say? Hello, my name is Carrie Smoke.